If you're able to, we recommend signing petitions or donating to bailout funds. Here's just a couple of posts with different organizations you can donate to or donating to the bailout funds, and we can send this out or post it on our Instagram. Other than that, we can get started with presentations.
All right, so I'll start off. Can you let me share my screen?
Yep.
All right. All right, so the article that I read about, it was about P53 mRNA and why it's important for cellular stress responses. So the reason why, so just a brief recap, the central dogma of biology, it's like one of the most fundamental things within biology, and it talks about how DNA becomes mRNA, and then it gets translated into proteins, and those proteins are important for how the cell operates and stuff. And RNA, as it turns out, actually has really important. Sorry, let me turn that down. RNA, as it turns out, has a really important history with how life came to be, and with how life started and regulation within cells. Okay, so this is just a little piece talking about RNA and how it's important with where life came from. Wait, can y'all hear this?
No.
Oh.
Unshare and share your computer sound.
What we know from good old, real, actual hard science.
Can you hear now?
Yeah.
All right, cool.
Today, no matter where we look in the tree of life, most cell machinery is made of protein, chains of folded amino acids. When modern cells make proteins, they copy genes from DNA into RNA and then use that RNA as a blueprint for making the proteins. We call this universal pathway the central dogma of biology because it sounds really cool and because it's something that all life shares. But there's a paradox hidden in there, a puzzle. It's a chicken and egg problem. DNA needs proteins to make more of itself, and cells need DNA and the instructions it holds to make proteins. So which came first? We can solve this paradox in a pretty simple way. Just get rid of DNA and protein in the earliest days of life, and let RNA do everything. RNA is the molecular cousin of DNA. It contains the same four-letter alphabet code as DNA, only T is replaced by a similar molecule, U. And instead of two strings in a helix, RNA is usually found in just one string. RNA is special because in addition to carrying information in that four-letter code, it can fold up into interesting shapes and actually do stuff. The same way that protein enzymes can do all kinds of chemical reactions, RNA enzymes, called ribozymes, can work life's machinery too. It's now thought that life began in an RNA world. Before DNA became a more permanent form of storage, different RNA chains could have carried information and been the machines for all of life's important chemistry. Unfortunately, the RNA-only world went extinct.
Okay, so that kind of gives you an idea of the importance of RNA. So the RNA that I mainly talk about in this presentation is P53 mRNA, and P53 It's known as a tumor suppressor. And basically how it works is, so like all of our cells have like a typical cell cycle. It starts off at G1, where the cell basically just grows, and then S, where DNA is synthesized for when the cell's gonna divide, and then G2 is when it's gonna get ready for mitosis by multiplying its organelles and stuff like that. And P53, it acts at those checkpoints in between the phases to make sure everything that was supposed to happen in those phases happened properly, and then it can carry on to the next phase. So P53 mRNA, it controls the RNA stability and the translation of the P53 protein that does work at those checkpoints. So what cells tend to do is that they take action on the P53 mRNA to make sure that the P53 protein is being synthesized at the rate that it needs to be and with the modifications that are necessary. So mRNAs, they have a certain type of structure. They start off with a cap at the end, and then they have an untranslated region and on both ends. And so the coding sequence is what the ribosomes read, and that's the essence of the protein. But then the UTR, the untranslated regions, at first scientists thought like, oh, this doesn't really have any purpose. It's just kind of there. But after recent studies, it's shown that the UTRs actually have an important role in stabilizing the protein expressions, the mRNA, and just controlling the response of mRNA translation. So this diagram at the top, you can see that at the 5' UTR, or the 5' UTR and the 3' UTR, all of these proteins, they take action on the mRNA. So for example, right here, the RPL26 protein, it increases the P53 translation whenever there is DNA damage. And it increases that expression by linking up with the 5' UTR. And studies have shown that there's actually like direct associations between different types of cancer and the mutations at these certain parts. So if you look at table 2 at the bottom, so melanoma, which is skin cancer, whenever there's a problem with the PTB protein, which attaches at the 5' UTR, there tends to be an association with melanoma. And outside of preventing DNA damage and making sure everything's synthesized, sometimes proteins also misfold in the endoplasmic reticulum, and that causes excess stress and stuff that can also lead to cancer. And so what the P53 does is it also reacts in response to that, and it tries to balance the newly synthesized P53 proteins with the older ones to ensure that the proteins aren't being misfolded. And whenever there is a misfolding, the P53 can essentially cause the cell to freeze, so that everything can be taken care of at the checkpoint, and then it can go on to the next phase in the cell cycle. Or sometimes if the damage is so bad to the point that it's beyond repair, it can cause apoptosis, which is, cell death. So because by causing cell death, you're preventing that mutated cell from multiplying and proliferating and maybe eventually becoming a type of cancer. This right here is an example of what happens when P53 is working properly and when there's DNA damage that's taken place. So at the top right here, this is on normal conditions. In normal conditions, if everything's worked normally, there's no DNA damage. The cell's all good to go at the checkpoint. MDM2, which is a negative regulator of P53, it's gonna attach to P53 and then it's gonna break down to P53 because the cell doesn't need P53 because everything's all good, there's no DNA damage, and so it's gonna break down to P53, and that kind of is a signal for the cell that everything's good to go, you can keep going. But if there is DNA damage, then this ATM right here, ATM is a kinase, and it's activated when there's a breakage in a DNA double strand. So it's basically like scanning DNA, and if it catches a DNA strand that's broken on both ends, it's gonna come and talk to the MDM2, and it's gonna tell it, yo, the DNA is damaged, y'all gotta make more P53. So the MDM2 is gonna get the message from the ATM, and then over here on the other side, it attaches to SER15, which is one of the spots on MDM2, and it's going to tell it up the P53, and it's going to make more P53 so that P53 can suppress and repair. And yeah, that's about it. So do you guys have any questions or anything like that?
Okay, if there's no questions, then I'll go next. So I did my okay. So I did my research on kiwi and how it affects the skin, because as you know, a lot of skincare products come out with a lot of different lines and they like to advertise different things. But last year, Kylie Skin came out and they had a face wash which really advertised like how it had kiwi in it and how it was so good for your skin. And I really want to see like what's the truth behind that. And so there was this experiment in this article, and it basically tested how kiwi fruit polysaccharides affect skin cells like keratinocytes, fibroblasts, and they even tested it on dermal equivalents. And fun fact before the actual results is that kiwi in Eastern medicine was actually known to treat different types of cancer because it inhibits it inhibits cancer cell growth, and it exhibits cell protection against oxidative DNA damage. And now getting to the actual results of the experiment. So what they did is when they took like kiwis from the fresh farmer market and they used different types of procedures to like extract the polysaccharides, and then they did different experiments on that, and I focus on 3 main ones. So the effect on human keratinocytes are basically found in adult human skin, and they form keratin. And when there was 1 microgram, there was no ribosomal protein synthesis. But then when they added like 10 or 100 micrograms, then there of like the kiwi saccharides, then there was ribosomal protein synthesis, which is basically when like the ribosomes role in cell turnover, so like the cell dividing. And as you can see from this chart, they looked at the mitochondrial activity in the HA cat cells, and with the kiwi fruit raw polysaccharides, the mitochondrial activity increased a lot. And this basically increases the amount of ATP produced, which stimulates cell growth. And then next, they saw the effects on fibroblasts. And fibroblasts are a type of biological cell that synthesizes the extracellular matrix, and collagen produces structural framework and plays a critical role in wound healing. And you see this used in a lot of plastic surgery procedures as well. And they looked at the proliferation rate, which is again basically how fast a cell turns over. And as you can see with the fibroblast growth factor groups, it increased to 180%, and then the RPS stimulation groups increased to 150%. So overall, kiwi fruit polysaccharides induced proliferation a lot. And then lastly, they actually grew these dermal equivalents with like collagen and other things that are found in the skin cells in laboratories for 28 days. And then at the end of 28 days, the collagen levels stabilized at a low level. And they used ascorbic acid as a positive control group because it was known to increase collagen production. And the skin equivalence level of RPS promoted collagen synthesis to about 100% more than the untreated control. So it basically doubled collagen synthesis. And at the end, they had these discussion paragraphs, and I wanted to break that down into terms that all of us could understand. So again, in the presence of RPS proliferation, so basically the ribosomes part in having the increasing the number of cells, increased was in the presence of RPS proliferation of NHK, HA, cat cells, and fibroblasts was significantly increased. And these are straight off the actual research article, so I wrote little notes. And then the fibroblasts are basically where collagen and other fibers are produced, and they're found in the skin cells. And then keratinocytes are where keratin is produced. And the basic of this paragraph was that kiwi fruits did improve collagen and keratin production. And collagen is important for your skin because it helps with the elasticity, and keratin is important because it helps with the barrier protection of your skin. And then this next paragraph basically stated that kiwi does stimulate activity of cell growth and increase metabolic activity such as mitochondrial turnover, which maintains cellular stability and prevents degenerative cells. And obviously those are good things. And lastly, based on the dermal equivalence, they found that even when there was little to none collagen being produced, kiwi fruit polysaccharides increase the production of collagen a lot, which again is good because collagen helps with the elasticity of your skin. And I included these diagrams to kind of show why these things were important. So as you can see in the younger skins, the collagen and the fibroblasts are a lot stronger and a lot more visible, whereas in the aged skin, there's not that much collagen there's not that much collagen, and the ones that are kind of like broken, and this causes wrinkles as time goes on. And then basically this experiment overall stated that kiwi is a good product in skincare, but again, it was just a lab experiment. It wasn't actually tested on real human skin, so you have to keep that in mind. And as well, they only tested on the polysaccharides in the kiwi, not all of the other areas. So if an ingredient does state that has kiwi in it, you have to really make sure what part of the kiwi and how they're using it as well. Yep, that's all for my presentation. So does anyone have any questions or anything? Okay. Do you have a question? What is it? I can't hear you.
I was muted. Does the article mention anything about things outside of kiwi? Like, what about polysaccharides and other fruits and stuff like that?
They didn't test. They didn't test any other polysaccharides. They specifically got kiwi from the farmers market, so fresh kiwi, and they just did test on that.
Okay. And do you think they're So say that the polysaccharides are good for your skin. Do you think there's other stuff in kiwi that like might counteract the polysaccharides?
They in the beginning of the article they did say that um, that if you use like parts of carbohydrates in kiwi, it could be very acidic. So there are some parts of kiwi that are very acidic, and that was like in the first two experiments. So I feel like maybe that could be negative. Obviously, anything super acidic for your skin is not good. So if that was included with other ingredients that were also like acidic, then I think it could be bad for your skin as well.
Thanks.
Okay.
Hey, can you guys see my screen properly? Can you guys hear me too? Okay. So I read an article that described the relationship between autism spectrum disorder and the gut microbiota. So for a brief overview of autism spectrum disorder for people that don't know, autism spectrum disorder refers to a group, a range of developmental disorders defined by non-neurotypical behavior, which is just behavior that you wouldn't find in the neurotypical or average person, quote unquote, and difficulty communicating and interacting socially. In 2012, it was estimated that approximately 15 out of every 1,000 8-year-olds had autism spectrum disorder, and more boys were more likely to have autism spectrum disorder than girls in that study. And it's super expensive to raise a child with autism spectrum disorder, which is relevant to this study because that's why the interaction with the gut microbiome is relevant. It costs almost 1.4 million US dollars to take care of a child with autism spectrum disorder in the United States. And a lot of those costs are associated with like specialized care, education needs, and the loss of parental productivity because they have to put more time into raising their child. And its relationship to the gut microbiome was originally observed because one of the highest like symptomatic comorbidities with autism spectrum disorder is constipation, but most GI comorbidities like flatulence, abdominal pain, diarrhea, they have a very high comorbidity rate with autism spectrum disorder, which led to scientists believing that the gut microbiome and the GI comorbidities play an important role in the etiology of autism spectrum disorder. And etiology just means like how autism spectrum disorder came around, what causes it to occur in humans, etc. And then so the next important question is, what is the gut microbiome or the gut microbiota? The gut microbiota just refers to like the communities of microbial organisms, fungi, bacteria, etc., that live within your gastrointestinal tract. It's shaped by many factors like birth mode, whether you had a cesarean section or you were just born through the vaginal tract, what antibiotics your mother used while she was pregnant with you, what your mother was eating, any environmental factors can affect it, and also, as you grow older, your personal diet and your personal antibiotic use and stuff will continue to affect your gut microbiota. Dysbiosis of the gut microbiota, which just means a microbial imbalance or a microbial defect in the gut microbiota, is associated with abnormal behavior, Crohn's disease, obesity, and IBD, which is inflammatory bowel disorder. And the gut microbiota, like measurements and dysbiosis, all of that is measured through usually stool samples, urine samples, stuff like that. So this diagram right here shows how a leaky gut can cause autism spectrum disorder. And what is a leaky gut? So basically your gut microbiome can influence your brain function through various toxins it produces that interact with your neurons, your immune system, your endocrine system, stuff like that. A leaky gut itself is when toxic neurons or toxic proteins are produced by your gut microbiome and then are released through the gut barrier into the mucosal cells of the stomach lining. These mucosal cells activate the immune response, essentially to come destroy the mucosal cells and the toxin. However, that immune response weakens your gut barrier, which leads to the leaky gut, because your weak gut barrier is now actually allowing the microbiota in the gut to get out of the gut and produce other toxins that can get past the blood-brain barrier and affect your brain, causing abnormal behaviors. This interaction here between the brain and the microbiota in the gut is what's like, this is sort of the axis on which the whole argument is based, that the gut microbiome significantly impacts, autism spectrum disorder and various other developmental disorders. So yeah. Does anyone have any questions on this diagram in particular? Because it's pretty important, and it's understandable that it's not like completely intuitive. Okay, so moving on. So as you saw in the diagram previously, the gut microbiota interact with neural pathways. So the way they interact with neural pathways is through a thing called metabolites. Those toxic proteins I was talking about earlier, those would be classified as metabolites. Metabolites are just the products of metabolic interactions. So they can be short chain fatty acids, various chemical compounds like phenyl compounds or free amino acids. And all of these can interact with the vagus nerve specifically, and the vagus pathway, which is a huge neural pathway connecting several organs to the central nervous system. And they can cause autism spectrum disorder through this pathway, or at least cause autism spectrum disorder-like behaviors. The short chain fatty acids I was talking about, they can also cross the blood-brain barrier if it is slightly impaired, which it normally is in individuals with autism spectrum disorder, and that can increase the effects of the short chain fatty acids on the central nervous system. And in mice models specifically, propionic acid was shown to induce autism-like behaviors in rat models and mice models. And specifically how it interacts with the neural pathway, it doesn't affect the neurons themselves. A lot of these metabolites target neurotransmitters like dopamine and serotonin, which are actually synthesized in the gut. And so when they're synthesized improperly, these neurotransmitters don't perform their function as well, and these neurotransmitters are super important. They're supposed to regulate motivation, happiness, motor function, emotional stability, learning ability. But when they're not synthesized properly, like they aren't in individuals with autism spectrum disorder, they can cause depression and effects resembling mental retardation, which is obviously a big problem. Another very important interaction, how the gut microbiome can interact with immune pathways. This is actually a real big part of how leaky gut actually occurs, which is, inflammatory cytokines go into the mucosa, and that initiates your immune system response, which destroys the mucosa, weakening the intestinal tract mucosal barrier, which is how the microbiota can get out and start releasing more toxins into your bloodstream. Another really important metabolite that's produced here is lipopolysaccharides. This can actually interact with your white blood cells and has been attributed to some minor autoimmune diseases and disorders that are also related to autism spectrum disorder that can prevent or inhibit, like biological development outside of neurological development. Like this could be why people with autism spectrum disorder have stunted growth or odd, growth patterns when compared to normal neurotypical people. So, this is the big takeaway from the article I read, which was, since the gut microbiota has such a significant impact on neural pathways and immune pathways in the brain, how can you use that to create potential therapies for children with autism spectrum disorder? And something that's important about all these therapies is they're way cheaper than the $1.4 million that it would take to, go through the supplemental learning and care additives that parents would need to care for a child with autism spectrum disorder. Probiotics, for those of you that don't know, a probiotic is just a term that's used to refer to any like positive quote unquote bacteria or healthy bacteria that can have positive effects on your gut microbiome or just your body overall. Some of these positive effects are like they can reduce depression symptoms, they can increase your metabolism, or some of these can even like start restructuring your intestinal barrier so that's stronger, or induce cell growth at a higher rate than it normally would occur in humans. And prebiotic therapies are just like probiotic therapies, except prebiotics that refers to the compounds in food that induce the growth of probiotics. So like you would have both therapies at once essentially, because the probiotic would introduce the bacteria to your GI tract, but then prebiotics would ensure that that bacteria could thrive there and grow and become part of your gut microbiota. Some examples of successful probiotic therapies is a Bacteroides fragilis was found to reduce gut permeability, alter the gut microbiota composition, and decrease autism spectrum disorder-like behaviors in rodent models. One of the big problems with probiotic and prebiotic therapies is that there have been no multi-center, large sample, randomized controlled studies. So we don't actually know if they would be, as effective as they appear in most studies in one of those large randomized studies, which is why, those studies need to occur so that we can find out, is this a viable solution for autism spectrum disorder? Some other potential therapies. Is there any other nerve besides the vagus nerve that also plays? Yes, there is. So the vagus nerve was the one I referenced specifically because the vagus nerve is just one of it leads to one of the largest nerve pathways in the body. But so you have a nervous, essentially like a web of nerves in your stomach lining mucosa that make up your, I guess we'll just call it the digestive neural tract. I can't remember the scientific name, but any of those nerves that connect to the outer body to the sympathetic nervous system can be impacted by, and any of the toxin produced by the gut microbiota. The vagus nerve is just the one that's most often targeted because of its proximity to the gut microbiota, and it's just very large, so you can get the most relevant data from that nerve. But yeah, any nerve could be affected by the gut microbiota, and that would then play a role in how it causes autism spectrum disorder. And for Christian, you asked, would this help to explain conditions comorbid to ASD such as bipolar disorder and ADD/ADHD? I assume you're talking about what I was talking about earlier with the how serotonin and dopamine production is affected. Yeah, that does it. One of the outside of like depression like symptoms and mental retardation like symptoms, it can cause bipolar disorder, ADD, ADHD. And these aren't the only neurotransmitters that are affected. Again, these are like relevant examples of neurotransmitters that are affected, but without a large randomized multi-center study being done on this specific topic, we don't know the extent to which the toxins produced by your gut microbiota affect neurotransmitters synthesized by that same gut microbiome. So the other potential therapies are fecal microbiota transplantation and microbiota transfer therapy. Those are essentially the same thing. Microbiota transfer therapy is a kind of fecal microbiota transplantation. And what FMT is, it's basically when the fecal microbiota from a healthy individual is delivered to a patient with, dysbiotic or an unbalanced gut microbiota. This dates back to 4th century China, where people used it to solve like diarrhea and stuff. However, it wasn't used as such then. It was associated with a lot of infections. Now it's been shown to be very effective in treating CDI, which is Clostridium difficile infections. That's just a type of gastrointestinal infection that causes diarrhea. It can be severe, normally it isn't. It's also been used to treat symptoms in patients with IBD and IBS. And while it's generally safe, the reason FMT specifically isn't the best therapy for individuals with autism spectrum disorders is that it can actually increase the adverse effects we see with autism spectrum disorder, like diarrhea, abdominal cramps, bloating. It can even cause fever and stuff, and that's not really something you want to add to an individual with autism spectrum disorder, because that shows that the gut microbiome isn't balancing. It's actually like interacting negatively. Are these therapies in use for children with ASD? Have they been tested yet? Probiotic and prebiotic therapies have gone through small term clinical trials, like level 1 clinical trials, but there have been no large multi-center studies done on them. FMT hasn't been used for autism spectrum disorder. It's used mainly for gastrointestinal morbidities, or, as we defined here, gastrointestinal comorbidities associated with autism spectrum disorder. But it's shown to effectively normalize the gut microbiota, and that's where like the study posits that the gut microbiota has such a significant effect on autism spectrum disorder that normalizing it would have, you would then reduce autism spectrum disorder like symptoms. The next one, microbiota transfer therapy, has been used in children with autism spectrum disorder. It was used in an open lab clinical trial, and what occurred basically was there was 14 days of antibiotic treatment used to, thin the herd in the gut microbiota, followed by a bowel cleansing, which again, just to remove as much of the unbalanced microbiota as possible, before you received a high initial dose of standardized human gut microbiota, and then that dose was continuously received in lower quantities for 7 to 8 weeks. This was used on children with autism spectrum disorder, and it was shown to be relatively effective. I don't know if this sounds right, but you can't cure autism spectrum disorder this way, or there is no, I don't know if you could return neurological pathways to functioning exactly as they're supposed to, or neurotransmitters functioning exactly as they're supposed to, but it has a significant impact reducing autism spectrum disorder-like behaviors, which is, which classifies as like a viable treatment. And finally, for dietary supplements and antibiotic treatments, various short-term antibiotics and, some unique diets like the gluten-free or ketogenic diets have been shown to improve autism spectrum disorder like symptoms, but they've only been shown to improve these symptoms in the very short term. So without, again, another long-term multi-center study being done to see how a dietary change or an antibiotic treatment would affect someone in, the years of their life that are yet to come, we don't really know if dietary supplements or antibiotic treatments on their own are enough to affect autism spectrum disorder symptoms, which is why the previous treatments, microbiota transfer therapy and probiotic therapies, are the ones that are, this study suggests that we look into more heavily because they've been shown to have longer term impacts. And the final slide I have is just, what I think are the most important takeaways. And that is that, current studies show that the gut microbiota most likely has an effect on the etiology of autism spectrum disorder. And that, current studies also show that the gut microbiome can affect your brain through neurological and immunological pathways. So what now needs to occur is we need research on the possible treatments that are being suggested. We need to try and normalize the gut microbiome in individuals with autism spectrum disorder and mice models with autism spectrum disorder to see if what the current mice model studies show us are replicable in humans, if we can reduce these symptoms, because it is a much less, expensive, and it's a much less invasive way to cure autism spectrum disorder as related to like autism spectrum disorder, behavioral therapy, learning supplements, care supplements, etc. And yeah, that's what my article is about. Do you guys have any more questions?
And I'm gonna be sharing, or Hudson, do you want to answer Shahbaz's question real quick?
Yeah. Yes, so the study does discuss this. So as you grow older, your gut microbiome begins to normalize because when you're born, right, your gut microbiome is mostly based on how you were born, cesarean section versus vaginal tract, because the bacteria found on your mother's skin versus your mother's vaginal tract are the bacteria that will make up a majority of your gut microbiome, along with the bacteria that your mother would have had when she was, whatever foods she ate and whatever antibiotics she ate or she consumed while she was pregnant with you. As you grow, however, you start to eat your own food and you start to diversify the food you eat as you grow older and older. And that stabilizes your gut microbiota normally into a healthy adult gut microbiota. So before your gut microbiota stabilizes and grows large enough that it's very hard to treat, like you need intensive colon cleansing, you would probably want to try and have these therapies occur then. So 8 years old and younger is what these therapies are normally targeted at, so that you can have the probiotics take effect while your gut microbiota is naturally growing and maturing. And that way, once you're an adult, your gut microbiome will sustain that probiotic bacteria, even if you don't stick with the treatment for your entire life. like just whatever you normally consume, part of that will go to that specific microbial community and help sustain it in your gut microbiome without any like additional effort on your part.
Okay. So the article that I chose to talk about for this week, it was conducted by researchers from the Netherlands, and they were specifically interested in this gene called the TSIN54 gene, and that's a subcomponent of a larger complex called the TSIN complex, and they're basically interested in doing mutations in this gene and modeling them in zebrafish to see what more they can learn about pontocerebellar hypoplasia, or PCH. So what is PCH? Well, it's a group of neurodegenerative autosomal recessive disorders, and it's characterized by a bunch of symptoms, the most notable being the fact that it causes severe mental and motor impairments. And the symptom that researchers are going to be looking at the most is the fact that PCH, it causes hypoplasia or atrophy of the cerebellum and the pons. And basically that just means that they're looking at how the brain kind of shrinks in response to these mutations and how it kind of loses its structural definition. So there's 11 subtypes of this disease. PCH2 and 4 are specifically caused by mutations in this TSIN complex. So this is the TSIN complex here, and this is the tRNA splicing endonuclease complex. And again, it takes those introns out of tRNA, and it's made up of 4 different proteins. So the 2 catalytic subunits are TSIN2 and TSIN34 over here. And these are the ones that kind of have like the enzyme activity that actually like cut that intron out of tRNA. And then the other 2 subunits are TSIN54 and TSIN15. And these are the structural subunits that just make sure that everything is lined up correctly. So in our cells, tRNA, it has a really important and essential role. So tRNA contains an anticodon that base pairs with the codon on mRNA, and that interaction ultimately helps generate a fully functioning protein. As I'm sure we all know, proteins are super essential in all of our cells. But how do we get that tRNA in the first place? So in your cells, you have some DNA, and then an RNA polymerase III comes along, and that creates a premature tRNA molecule. And then the TSIN complex comes, and it cuts that intron out, and it creates these exon products that then get ligated together to form the spliced product. And that spliced product goes through other modifications that ultimately forms that mature tRNA molecule. And again, I just want to emphasize that mature tRNA molecule is made because that TSIN complex is one of those preliminary steps in ultimately generating that tRNA. So the researchers were specifically focused on TSIN54, again, which is a subcomponent of the TSIN complex. And they're hypothesizing that this disease is caused because the TSIN54 gene cannot function properly. So kind of like an overview for all of the experiments is first, they're going to start by knocking the gene down. And that just means that they're going to reduce the levels of that protein present. And they're going to see, does that cause a PCH-like disease state? So that's what they're really focused on. They're focused on knocking the gene down and seeing what that does to zebrafish brains. And kind of like the overall question that they're trying to answer is figuring out if there is a common disease pathway between TSIN54 and other PCH-related genes. So in addition to looking at TSIN54, researchers are also going to be conducting the same experiments on RARS2. So RARS2 is another gene that's also involved in RNA processing, and it's also been implicated in causing other subtypes of PCH. So their thought process here is that if they do the same experiments on both of these 2 genes, they can analyze like the similarities and differences between the results to figure out if they are acting through kind of like the same mechanism. So before they can even begin knocking genes down and seeing what happens, they first have to establish like the expression pattern for TSIN54 in the brain. And on the left is 8-week-old human fetal brain sections, and they marked them with a TSIN54 probe to see where TSIN54 gets expressed in the human brain. And when they did that, they noticed that the mesencephalon and the telencephalon, which I've highlighted here, those have the highest regions of TSIN54 expression. And the reason that that's important is because the telencephalon and the metencephalon, they grow to develop the pons in the cerebellum, and that's the areas of the brain that's most impacted by TSIN54 deletion in PCH. So that's the area that the disease impacts the most. So they did the same thing over here on the right with the zebrafish brain. And when they looked for the TSIN54 expression, they found that it's the highest in that telencephalon region and the mid-hindbrain region. So in all of the experiments I'm going to show you guys after this, just be on the lookout for the mid-hindbrain region and how that changes here with TSIN54 present versus what it looks like when TSIN54 is gone. So these are the results of after knocking TSIN54 down. So up top, the A and B are both the controls. So A is just the wild type, nothing's been done to it. And then B is like the morpholino just on its own with nothing else. So that's kind of like the negative control. And as you can see, that mid-hindbrain region where the asterisk is, that's like really well developed, everything looks normal. And then C and D are both the results of the TSIN54 knockdown. So when TSIN54 expression is reduced, you guys can kind of see how like that mid-hindbrain region, how that shrivels up and it looks like very sickly in comparison. And then the bottom two, E and F, those are the rescues. So that's how the embryos look after being injected with human TSIN54 mRNA. So those are kind of comparable to the controls on top. And as you can see, they're nowhere near as sickly as the embryos C and D look. So this part of the experiment, it was really important in showing researchers that knocking TSIN54 down, it leads to a loss of structural integrity in the brain. So kind of going off of that experiment, researchers, they took cDNA from those embryos and they did a reverse transcriptase PCR. And basically what that means is that they took all of that DNA from those embryos of interest and they kind of like turned it back into RNA and they ran it on a gel to compare and contrast, which is right here. So the wild type and the co-MO, again, those are both of the controls. So that's what we expect to see in normal cells. And the interesting thing is right here. So that last lane is that TSIN54 splice morphine. So that's like, that's the product length after that TSIN54 was pretty much like knocked down a lot. And as you can see, those embryos, they had a very, very short product in comparison to the controls. And the reason that that's important is because it shows that abnormal splicing is occurring, which means that the TSIN complex is not functioning correctly when that TSIN54 gene is present in minimal amounts. So along with like all the stuff that I just showed you on the previous slide, researchers also quantified those phenotypes into that bar graph here. And so they were qualified as being either normal, mild, or severe. So the first two bars are just both of the controls. And as you can see, it's what we expect to see when TSIN54 is present. It's mainly a normal phenotype. But the interesting thing is in the last two bars. So the third one is the TSIN54 knockdown. And as you can see, the dominant phenotype is severe. And then it's an interesting contrast to the last bar. So the last bar is that rescued embryo. So it was injected with TSIN54 mRNA. And as you can see, it changes from being a severe phenotype to being more of a mild or a normal phenotype. And that was important in showing that if you add TSIN54 back into those embryos, it can partially rescue that phenotype. So it'll reduce those harmful effects of TSIN54 not being there. So then this part of the study was focused on figuring out if there were any like brain developmental patterning defects associated with the disease. So in order to test that, they labeled the zebrafish embryo brains with 2 RNA probes. They labeled them with FGF8 and OTX2. So these are just RNA markers that show normal brain development. So the thought process here is that if these probes localize to a different part of the brain in different treatments, then that means that there's abnormal brain development going on with the TSIN54 knockdown. So everything on the left, A through F, is focused on FGF8, and then everything G through L on the right is focused on OTX2. So A, B, and C are just a lateral view of the treatments, and then D, E, and F are the dorsal view, and that's the same thing on the right, lateral and dorsal. So across the top is just the control, so that's what the localization of those probes look like when TSIN54 is present. And then the middle row is what it looks like when TSIN54 is knocked down. And then the last row is showing what that localization looks like when the TSIN54 mRNA is injected, so like for the rescues. So as you can see, there's not a lot of difference anywhere between any of these treatments or between any of the probes. And that was important because it let researchers know that knocking down TSIN54, it doesn't cause a brain developmental like disorder, that everything else develops fine. It's just for whatever reason, the TSIN54 and the tRNA are the only things being affected. And that led researchers to wonder like kind of like what the levels of neurodegeneration are in those cells that have TSIN54 knockdown. So in order to study that, they incubated the embryos with acridine orange, and that's just a dye that binds to the nucleic acids of damaged cells, and it causes them to fluoresce. So essentially, with that dye, if there are any damaged cells, they'll fluoresce, and that'll be a signal for the researchers to know that there are levels of neurodegeneration in that treatment group. So A and B are both of the controls, and as you can see, there's like barely any fluorescence present. But the interesting thing is when you get to C, so C is the treatment that has that TSIN54 knockdown. And as you can see, there's so much fluorescence here. And that's a really good contrast to the treatment D. So treatment D is the rescued embryo. So it had the knockdown, but they injected the TSIN54 mRNA back into it. And as you can see, there's very low levels of fluorescence when it's added back. And so all of that was also put into this bar graph here. And as you can see, like the most striking part of it is that TSIN54 knockdown and just how high the levels of fluorescence and neurodegeneration are. So this part of the study was really important in letting researchers know that knocking down TSIN54, it does lead to high levels of neurodegeneration in the cells. And so everything we've been talking about, like the knockdowns and stuff, that's kind of like a temporary look at what's going on because those effects, they go away after a few days. So to kind of account for that, researchers, they wanted to come up with like a more long-term model to look at mutations to TSIN54. So what they did was they created a strain of zebrafish that had that premature stop codon for TSIN54, which essentially like deletes the gene. And then they crossbred a bunch of other different zebrafish strains until they had a strain like colored in yellow that was homozygous for the stop codon as well as heterozygotes. And then they compared that to the wild type as well. So what we're really looking at here is this yellow line on this graph. And as you can see, after 9 days, no zebrafish that had that stop codon that were homozygous for it survived. And that was really, really important in showing researchers that for whatever reason, TSIN54 is essential for life. And so everything that we just talked about with TSIN54, researchers also did those same experiments with RARS2, with the RARS2 gene. And as you can see, it kind of had similar effects. A and B are the controls that looked healthy. C and D are those morphins with the RARS2 knockdown. And as you can see, when you knock it down, that mid-hindbrain region is affected a lot, kind of in the same way that TSIN54 was. And then E and F are the rescues that look really similar to the controls. And then again, they did the same reverse transcriptase PCR experiment. They quantified everything just the same way again. And as you can see, the controls, the product was shorter, which meant that abnormal splicing happened. And also, both of the controls had a normal phenotype. But when you knock down that RARS2 gene, you have mainly a mild or a severe phenotype. And again, it's rescued by adding RARS2 mRNA back in, which causes it to change to a mainly normal phenotype. So the main takeaway from doing that was researchers, they noted just how similar TSIN54 and RARS2 knockdowns were. And that's kind of like indicative of like a common disease pathway. So by doing all of this, researchers, they saw how inhibiting TSIN54, it did lead to neurological abnormalities and that the brain was not nearly as properly developed as the control embryo was. And that kind of led them to confirm their hypothesis that TSIN54, it's probably happening through a loss of function mechanism. And another interesting thing was that there was no brain patterning defect associated with PCH. And they were also able to see the high levels of neurodegeneration caused by knocking down TSIN54. And then also just the similarities with RARS2 and Tsin54, just because they're both involved in RNA processing and they cause the same disease, that kind of led like researchers to think that maybe Tsin54 and RARS2, maybe they have like an alternative function that we don't know about that could also be causing the disease. So that's like an interesting avenue for future research. Do you guys have any questions? Oh, they're focusing on the Tsin54 gene because out of like that entire complex, like the Tsin complex, that's the one that usually suffers the most mutations. And that's also the one that causes like the most severe forms of PCH. So that's why they focused on that gene specifically. And also like there's just not a lot known about this disease in general. So that's why they're kind of trying to model it in zebrafish and see like exactly what effect that gene has on like the development of the brain and stuff like that.
Okay.
So putting human Tsin mRNA in zebrafish, does it cause anything weird to happen because it's a different species? And also, why were zebrafish chosen as the model species? So actually, like a lot of the time when you're modeling disease, researchers, they usually use zebrafish just because they're super easy to work with. They have like a really low, like they reproduce really quickly. So if you mutate them, you'll usually like see those effects really quickly. So that's why even apart from like the article that I chose, usually if you do disease modeling, it'll usually relate to zebrafish because of that. And also another reason is the zebrafish genome is very similar to the human genome. So like a lot of the diseases that cause like problems in humans, there you can find like an analog or like a similar gene to that in zebrafish. So that's why it's also really similar because you can like see a more direct relationship in zebrafish that have like the mutation that you're looking for. And human Tsin mRNA, I don't think it causes like a weird effect to happen just because of how similar like that DNA and those nucleic acids are. If that answers your question. Currently, I don't think I know of any mammalian models that have been involved. For me, everything that I've looked up about the Tsin complex and PCH, it usually comes back to like disease modeling in zebrafish mainly, just because it's so easy like to see that pattern displayed, and it's just easier to study. There's more info about zebrafish than any other type of mammalian model.
So if anyone has any questions overall by anyone's articles or presentations, please feel free to email STEM Journal Club or go on our Instagram. And we even got the blog set up for you guys. So here's the basic overview, and we will be sending this out in a link so you guys can obviously access it. It's easy, it's through WordPress. And so I just created like a sample post so you guys could kind of see. So the post will probably be here. And I really want to emphasize that this blog is not just for NC State kids. So if you have research and you present it and you want to share it with like after you present it with us, you can definitely share it with more people, such as your friends or family, to show them your research, or even professors in the future if they ask like, is this like written anywhere, do you have any anything to show, then obviously you have this blog, you can show them. And it's super easy to use. So basically all you do is you click add write, and it will let you add a title and then whatever you want. And I had basically like mine written up so you guys could see. So this is all I wrote for the actual blog, and I included my article, and I just saved my Google Doc as a PDF and included that as well. So I looked it up, and sharing the videos that we present, like if you want to attach a video, it costs money, but I can maybe find a way to post the videos people present on YouTube and then just attach that link so people can also see your presentations if you guys would like that. So I just wanted to mention that the blog is up and everything.
And another thing I want to say is thank you guys for coming out. It means a lot to have you guys support us. And I know we may have confused you guys with like the group emails and stuff like that, but our thought process like for putting you guys into the smaller groups was just because we had like over 100 members, and we really wanted to make sure that each person got to talk about something that interested them. But seeing as how only 30 or 35 people showed up to this meeting, we'll go back and like adjust the group size accordingly, like either making it one group or two big groups or however that works. But if you guys have any questions about it, or if you guys have any suggestions on what we can do better to like better reach out to you guys, please let us know. Like we're open to like any suggestions that you guys might have. So
Yeah, if you have any questions about the groups and stuff, this would be a great time to ask. I know we got a lot of emails saying that the group invitations had expired. We didn't know that they expired, but we can resend those out so that if any of your friends or anyone you know wants to get in and they're like, well, we don't know how, we can just send them out. And if you know anyone who's interested and you want to get them involved, just tell them to send us an email, or you can forward us their email and we can reach out. So it's fine. Why are you laughing?
the typo on NCSU.
Oh, I didn't do that. But we can fix that as well.
Yeah, thank you guys for coming. And if there's anything else you guys like need, or if you guys have any questions, just feel free to shoot us an email, and make sure you guys follow us on Instagram because we always like post up-to-date content on there as well. Thank you guys for coming.
Also, please sign up for next week's presentation, and we'll also be having a guest speaker next week right after the presentation. So from like 2 on, and it's Dr. Hartstone.